We're meeting at 2:30, right, for the next one?
Today.
Yeah.
Yeah.
And then, is it just at your office? Okay.
I got a flat tire this morning and they couldn't come to the garage. Is there any way I can go to the
We got one 10.
Wednesday? Can I come up?
Thank you.
Well, I'm gonna try to get all these graded maybe this afternoon. I'm trying. Been in meetings yesterday and all day tomorrow, so, but I'll get them done. So, don't forget, you don't have to show up. Friday's reading days. In case you don't know what reading days are, every year, it's set up almost like a spring break, almost, for students. Fall break. But, faculty will be meetings and doing things like that, so it gives the time to do those, but you guys don't have to show up. Any of your classes, not just this one, so you're done after today for the week. And I will get those, we'll have those exams back for you Monday. And I'm going to download the new chapters. I don't know how anything went on those yet. I've not been through those, but, we're gonna get in on some things right now. It's gonna start to pick up a little bit, get away from the chemistry. And we're gonna be getting into more the biology, particularly at the cell level. Talking about some very important things. Now, the end of this course gets kind of busy because we start talking about some heavy-duty things like genetics, heredity, cellular respiration. So now is the time. Make sure you take good notes. And keep up with what we're doing. So I'm going to download here these new units. I'll pop it right back up on the screen and you'll be able to see.
Chapter
7. We'll start with that, yeah. There's some of these that in this first part we don't cover as much. There's certain parts to it, but we just don't cover as much as we do on some of the others.
So will we have 3 chapters again and then our next exam?
Yeah. These, it depends on the or the chapters. Some of these I like to break up because the information may not go as well together as others. And I really don't like to mix too much of some of this. So we will have chapter 7, 8. And we'll take a look at these and make our plans right now. Now, we gotta be kind of careful here because we're also gonna be popping up, but this, they say there's 14 weeks in this semester, I don't believe it. You know, you'll miss one Friday, you'll miss one Thanksgiving. So that's almost a week right there gone, and, I but it just seems to me that, it picks up, is what I'm trying to say. And, there's gonna be a lot of material, and the complexity of that material will increase. Unit 7, it'll pop up on screen here in a minute, just trying to warm up. It's gonna be about the membrane of the cell. There's gonna be some important things in it, but we're not gonna spend forever on it. So this is one, pay attention to the parts that I'm telling you to remember. There's a lot of things in the cell we're not gonna take the time to cover, we gotta cover other things. And it's a big book, there's 56 chapters in this book. Normally, both sections will get through maybe 30. There's a lot of chapters that are just really not for this course, it's for other courses that's in there. So, this one is membrane, chapter 7 is the membrane, so we will pick certain things in that. Then next up is the big one. This is going to be that chapter starting right there, metabolism, would probably be the one that most people consider to be the toughest. A lot of material, a lot of technical material. It will lead into cellular respiration. So by the time we get done with cellular respiration, there's gonna be a bunch of material. That's another reason I don't want to spend too much time on the membrane. Yeah.
We're starting on that,
Do what?
we're starting on 9, not 7.
No, we're starting on 7. I'm just showing you the ones that's coming.
Oh,
So 8 is gonna be about energy in general and how it works in organic systems. And this is gonna be the big one. And this is gonna be how your body and all animals' body uses energy right down to every electron whenever you eat food. So there's a lot of information in there, which is, as I say, another reason we are not going to beat the membrane to death. I'm gonna pick out certain things with it. We're gonna move along because it's gonna take some time for the other stuff. So we will start with 7. We talked a little bit about these organelles the last go around, just as an overview. And now we're going to get into specifics. The membrane is extremely important because it controls the environment, the internal environment of that cell. That membrane is going to control what comes in, what goes out. It's a gate, a wall. Still my clicker's not working. So I guess I have to stand over here. That part works. As I say, the membrane controls what comes in and goes out. That is called selective permeability. Those little two lipid membranes that you drew with the phosphate head and the lipid tails, right there. Selective permeability controls what goes in and what goes out. Phospholipids are what make up that membrane. Here's a new term that you will hear in many other things in biology, not just concerning this. Amphipathic. It contains both hydrophobic and hydrophilic regions. So this is a molecule that's not going to be either hydrophobic or hydrophilic. It's going to be both in different areas. Amphi is going to mean both, and the pathic is referring to the facts and likes and dislikes. So remember, amphipathic molecules. These membranes are going to be, if you turn right here, it says the fluid mosaic model. And it calls it a fluid mosaic model because there are a lot of things in it. It is a mosaic. It's different things that come together to make the full picture. And this just is the basic membrane with nothing added. Remember the phosphate heads, hydrophobic tails made out of two fatty acid chains. They are going to be immersed in water, whether it is outside of the cell or inside. Now, they're taking that same membrane and starting to put proteins in it. Notice here, in that protein, since it's going to be partly in fat, and that part that's going to be in fat is going to be hydrophobic, right?
Mm-hmm.
The part that's going to be associated with water has to be hydrophilic. So that's where that molecule comes from. These special proteins in here, hydrophobic, on the outside, hydrophilic. That's where that term amphipathic comes from. Now this just shows these as solid proteins, and they can be solid. They can also have channels that let things come in and out. Proteins are the big communication device within a cell. Some of these proteins can have receptors on them, and whenever that something matches up with that receptor, it will send a message all the way through that protein to the inside of the cell telling it to do something. So we will look at the different kinds of proteins. One of the ways they study these particular lipid bilayers is called freeze fracture. They actually will freeze the cell and take the two lipid layers apart. And that's what it looks like. That way, they can actually take out those proteins, isolate them, and figure out what they are. What do they do? So, freeze fracture is just a way of separating the layers. And these are some little things, like I said, we're not going to cover an enormous amount on this chapter, but here are some of the ones to remember. These phospholipids that are in that membrane can move. We showed them together as those little tails and so forth. They can move and switch places. Notice it says most of them move laterally across. In other words, you've got one here and one here. They can flip and go to different places. Sometimes you can have them like that, and then one can actually flip over and go on the other side. It doesn't happen that much. There's reason for all of this. This is more of what a membrane would look like if we actually looked at it under a microscope. Notice there's proteins going through, but there's different kinds, different shapes. Notice these filaments and things that we talked about that give structure to the inside of the cell. And notice right here, cholesterol. Cholesterol is in that membrane. Now, people who like to eat chicken and things like that, they'll tell you to take the skin off because that's where the cholesterol stuff is. But there's an example of it. Now, the reason that there is cholesterol is something to remember. cholesterol increases the fluidity of that membrane. That means if you're in cold weather, things start to chill, whereas fat normally wants to congeal and get dense. Cholesterol helps to keep these fatty acid tails from compacting. It won't let them compact and get more dense, so it maintains fluidity. Notice these proteins, these little ends. These are going to be the receptors. Notice how it's going up here to this. It's going to be the branch of the nerve. And notice how that protein will go up. It will make a connection with that nerve, and now we have a path where we can actually send messages from your nervous system right into the cell. Notice the green that comes out of these proteins. When you have those greens are going to represent a carbohydrate. When you put that carbohydrate with one of these proteins, it's going to have a particular name to it. If we had a fat attached to it, it would be a lipoprotein. And this is called a glyco, referring to the sugar, glycoprotein. Glycoproteins are the one that have the receptors that enable your body to scan over them to determine is that your cell or not. They can do other things and send other messages. That's just one. But these glycoproteins are going to be involved in communication. Notice there are other carbohydrates by themselves not attached that can also do things. Different kinds of proteins. They come all the way through. They can be a trans protein or integral. If they're only on one side, they're called peripheral, meaning to the side. So notice up here, all of these different little structures, cholesterol, the membrane, and so forth. Cells have to communicate with the outside environment. Your body has to be able to send messages to that cell and get it to do something. If a lot of sugar is in your blood, glucose, and it starts to attach to some of these receptors, down in the pancreas, it says, sends a message through these, start making protein, or the insulin. That's what it is. Nerves up here, if this were muscle cells, you can tell it contract. So there's a lot of things. Everything basically is going to have some communication to it in one form or another. This shows the movements, how they can just simply move side to side. They do it constantly. It's just a way of maintaining fluidity. And here's why. It says as temperature cools, the membrane switch from the fluid state to a solid state. The temperature at which a membrane solidifies depends upon the types of lipids. Membranes rich in unsaturated fatty acids are more fluid than those in saturated. Remember, if they're saturated, they're kind of packed together. But if they're unsaturated, they get dense, they got more space and more room to move. Membranes must be fluid to work properly. And it's, if you notice here, it says they normally are about as dense or about as fluid as solid oil. So even though we're thinking of these as big molecules, if we were to cut open a membrane, it would be more fluid-like than solid on the inside. Steroid cholesterol has different effects on membrane fluidity. Warm temperatures such as 37 degrees Celsius, remember room temperature would be about 20 to 22, depending on how cold you want a room. Human body temperature is 37. So in human body temperature, cholesterol restrains the movement of the phospholipids. It doesn't let them move too much. It's kind of a barrier there. So they can move, but they can't move too much. At cool temperatures, it maintains fluidity by preventing those little things from getting condensed, shrinking. Notice the fluidity, unsaturated hydrocarbon tails, saturated tails. So this is going to be a membrane that's fluid. That one's viscous. Viscous means thick. Boy, I have the green thought I saw some.
So will we be needing to draw any of this so far?
No.
Oh, thank you.
I'll tell you.
Thank you.
Now notice down at the bottom, this shows the cholesterol that's in here. That helps to prevent these from packing too much. And remember, when things heat up, motion increases, so it prevents them from getting out of hand, messing the membrane up as well by too much movement. The only thing you really need to remember, what's the role of cholesterol? Remember, as the membranes start to become colder, they become more viscous, more dense. Cholesterol helps to prevent them from being packed and then becoming so dense that it loses its ability to function. Talking about proteins, now this you should remember as well. Peripheral proteins are bound to the surface of the membrane. They don't go all the way through it. Integral proteins penetrate into that hydrophobic core, the fatty acid tails. If those integral proteins go all the way through, they are called trans, which means across membrane, all the way across the membrane, transmembrane. And the hydrophobic regions are nonpolar, like we said.
So these integral proteins, they penetrate the hydrophobic core, but they may not always go all the way through, right?
Correct.
So sometimes when they do go all the way through, they're called
Transmembrane protein.
transmembrane. Thank you.
And how do we get those polar, non-polar regions? By the makeup of the amino acids that make up that particular section of the protein. Notice it says they're alpha coiled into alpha helices. So look right there, and that gives you a little kind of a breakdown. These are the non-polar regions, or the polar regions. These are the non-polar. Notice on the inside where we have that polarity, here, here, and here, there is no polarity. These are the ones that will be hydrophobic, so those sections are going to be next to the fats that are also non-polar, and these polar areas will be next to the side where the water is that will be polar. And still, we have those N-terminuses and C-terminuses just like this before.
So why exactly is the inside of the amino groups, I mean, the secondary structure, why is it non-polar, but the outside ends are polar?
So this fat that it's in is non-polar.
So that automatically makes it non-polar also?
Well, if you had something that was polar, these push each other away, they don't mix.
Right, so there, I was just thinking
It
if,
has to be compatible. The reason it's non-polar is to be compatible with fatty acid tails.
okay, so the reason that they're there is because they're compatible, but is it because of their structure that makes them compatible?
The amino acid structure. And remember, proteins, we talked about primary, secondary, and quaternary. As you can see right there, these are many strands, a lot of them of these secondary structures that you see, the helix structure. Six functions of the proteins you should know. One is transport. They can let things in or out They can act and be involved with enzyme activity. Signal transduction, sending messages, receiving messages. Or this membrane and the proteins help cells to recognize each other as being normal because of the protein structure. These membranes can also join to make tissue. Intracellular joint. And they can attach to the cytoskeleton and the extracellular matrix, all that stuff on the outside of the cell. So these proteins, you can have fibers and things going through, and those cells can actually attach to it and have an anchor to where they got something basically to hold on to, and that helps in the tissue formation and so forth for strength. Shows you some examples of each of those. Transport, just let things in and out. Enzyme, they can act like an enzyme, where other things come and attach to the enzyme and activate it. It can act as a receptor. Some kind of molecules produced, like we mentioned, maybe glucose, and tells it to do something else. But that's what I said. Transport, it can open up, have things in it, and let it go through another one. Just simply because it can close on one end, open up on the outside end, let stuff in, close, and then open up on the bottom end and let it through. This is the intercellular joining, the way they can come together to make tissue. And this shows how they can anchor to the cytoskeleton, whether it's nerves or whatever it is. This is very important, this cell-to-cell recognition. Remember, your immune cells have to be able to tell that the cells in your body belong to you, so the immune system does not attack them. And they also have to be able to tell if something like a bacteria gets into your body, they have to be able to recognize that it is foreign, so they can go through their process of getting rid of that foreign body, the microbe. And if you look, it says membrane carbohydrates are covalently bonded to lipids called glycolipids, or more commonly to proteins called glycoproteins that we mentioned earlier. Carbohydrates on the external side of the plasma membrane are vary among species, individuals, and even cell types. You can use things such as the composition of the protein in the carbohydrate, for example, to determine what species of animal it is, or even if the cells came from the same individual. How do you think you can figure out what the amino acid structure of a protein is? Do what? Too complicated. Big machine, hooked to a computer. We want to know if I had a protein upstairs and I wanted to know what its amino acid sequence was, I can run gel electrophoresis to isolate that protein, cut it out, send it downtown, university to a lab. They'll take those proteins, basically mix it with a few little chemicals, clean it up, denature it, put it in a big machine hooked to a computer, give it 24 hours, then it'll come back and tell you the amino acid sequence, one by one. So, when you have the computer software that can tell machines what to do, for the most part, we can do it for yeast, and you can do that also with genes for DNA. That is called sequencing. And this would just be amino acid sequencing for protein or nucleotide sequencing with genes. Yeah. Do you have a question? Oh, I thought you raised your hand. So, you know,
So should we memorize this? Like
No.
this? Okay.
you need to know what a glycolipid is and glycoprotein is. And know Notice it says cells recognize each other by binding to surface molecules. They contain carbohydrates on the extracellular surface of the membrane. They're also going to be attached now to proteins, but usually they will be glycoprotein. Now this is AIDS. Now notice how AIDS works right here. HIV is the virus. It is called human immunodeficiency virus. That's what HIV stands for. It makes your immune system stop functioning. How does it do that? The reason your body can't cure AIDS is because when you get the virus, it lives inside of your own cells. So when your immune system comes along and sees the cells that's there, it's your normal cells. It doesn't recognize that the virus is living inside. So what happens? Along comes the HIV. This is the virus. The red stuff in through here are proteins, and I think the green stuff represents the enzymes that's going to help it to get in your system and so forth. Look at what happens. These are the proteins on the outside. All of our cells have certain receptors. These red ones here are called CD4 type receptors. HIV has the ability, whenever it has the CCR5, these two receptors together. Notice how the little round shape that it has right there. This little round shape and how well that fits into that little spot. It's like a lock and key. So what happens? Whenever these proteins that we see up through here, it's just like you put a key in a lock and then turn it. What happens? That membrane will then allow, not the whole thing, this membrane sac that it's in, but it will allow the main part of that virus then to just be absorbed and go right into the cell. Based on receptors. Now look at the other side. There are some people, and many of these are individual ethnic groups, it seems, and it's genetic, inherited on whether you have both of those receptors. Some do not have those receptors, and the HIV virus, even though it's there, cannot penetrate the cell. People will never get AIDS because they can't pick up the virus because it's lacking that CCR5 receptor. There's a lot of work being done on trying to make vaccines and things to maybe create that pseudo nature wise. In other words, we do it in a lab and give you a shot of vaccination so it prevents this binding from occurring. And if we can do that, then you won't get AIDS.
So basically, to put something into the binding site so that the HIV cannot bind to it. But does anything else bind to those specific receptors so that it could interfere with something else?
You know, that's why they're there, to do a lot of
Right.
different things. I don't know exactly what they are. You've got tons of receptors on your cells that do, I have no clue how much. But yeah, they got a function. That's what a virus or a microbe does. They take advantage of what you've got and they change themselves then to match up. That's adaptation. What it is. Yeah.
So if you didn't have
Yeah.
the CCR5 and you got infected with HIV,
You
would
won't.
it still be in your system and
No.
would you give it to someone else then? No?
It would, your immune system would have enough time to take care of it. AIDS, folks, and believe me, I've done a lot with it. People in the last dying stages of AIDS, people contaminated with HIV, I've worked in their blood and tissues, or it's not as easy to get as what people think. One of the most dumb things that you'll ever see if you're ever watched, like on in a basketball game now, they have rules. If anybody gets a bloody nose, if they don't have a bloody nose, it's not a very good basketball game. But if anybody gets a bloody nose, they gotta stop the game from pouring bleach out, wiping down the court, making them change shirts, and it's stupidity. You're not gonna get AIDS that way. It don't happen like that. If we had somebody that was contaminated with HIV, and you had a cut, and we took that blood and poured it into that cut, you're not gonna get AIDS. There's been, when AIDS first came out, the first thing they done was a study, and it showed out of 100 instances where that actually happened with hospital workers, not a single one ever came positive. And there's a reason for that. Many of these microbes have to be in the correct environment. Now, if HIV is exposed to air, it is extremely difficult to get. It's not that it's impossible, but it's been estimated, if you take like a regular syringe, about like that's about 1 cc, it would take 1 cc of blood that's been exposed to air, injected into your body for you then to become HIV positive. However, if it's something like the lumen of a needle, different story. It only takes small, very small amount. There can be more than enough inside the lumen of a needle because it's not coming in contact with air then. If you, and this was what happened with the first thing that AIDS really took off on was the drug addicts that were basically shooting up heroin at the time. And so they would be putting needles in and then sharing needles with each other, and there'd be enough blood in there, you got it. A little boy in Indiana died named Ryan White when AIDS first became known because he got a blood transfusion from someone. The drug addicts would often go in to the hospital and donate blood to get money, and this little guy got it. He was like 10, 12, and died from it before anybody even knew what it was.
Hemophiliac at first, and then you got the.
So, you know, if it's not been exposed, you can get it. Now, it's been isolated, things like tears, saliva, but I know of no instance where anybody ever like drank after somebody and got it. Now, if you were to put that saliva maybe in a syringe and shot it into them, maybe, but it's not that easy.
Is that why why needle sticks are so serious? Like
There's one virus out there that is unlike AIDS, where AIDS is difficult to get. This one, you can have 1 drop of blood on a tabletop, dried up, get it in a cut, and you can get it. That's hepatitis B. And all of the health universal precautions are, if you can prevent yourself from getting hepatitis B, you ain't never gonna get AIDS in a hospital setting anyway. So, if you have a needle stick right now, it has to be reported to the health department, to agencies, all over. I've been stuck with them too.
have you ever reported?
Oh, yeah. And I was taking care of a mission hospital and worked on a patient, done surgery on them, never got stuck. Turns out that patient had HIV, so you have to wait 6 weeks from the time that you're infected or could have been infected before you can do the test to find out if you have been infected. It takes 6 weeks for your body to develop the antibodies and the proteins and stuff that they test for, called Western blot.
I know they're actually saying that garbage and sanitation workers could be at risk of all the needles lying around with the heroin.
That's a big deal, and that's why, like, if you go up in our labs, we've got the hazardous waste bags. If you ever looked at them, they're really thick. Now, they're not gonna prevent scalpel blade or anything, but on the walls, you've got the little sharp spots.
Like you see in the
They're
back.
designed to where some of them you just cut the needle off, some of them you throw the whole thing in the blade, and that's why they're all up there. But getting a puncture wound from something like an old syringe, the kind you see laying out in the street, absolutely you can get it. So,
Do they test blood? I mean, people who give blood, like you said,
They test it now, you better believe me. You're not gonna get it from now, it's not gonna happen. But whenever this happened to that little guy, and it happened to other people too, he was basically the first one that people really noticed. They weren't checking because they didn't even know the bloody thing existed. And, so now, yeah, they check it very well. So, there's a lot of research being done on HIV and many other diseases, and a lot of it focuses on these proteins that are in the membrane and how they act as receptors. Can you block some of this stuff with vaccines? So, notice it says membranes have distinct inside and out phases, which means they don't look the same, obviously. Asymmetrical distribution of proteins, lipids, and associated carbohydrates in the membrane is determined when the membrane is built by the ER and the Golgi. Notice glycolipid filled in that little, all of these glycolipids right here. That's the protein, where the carbohydrate part, there's the lipid parts that come together. Your body can make these little vesicles from the Golgi can then just pinch off, go fuse right through the membrane, and release, as you see, the secreted protein or the glycolipids and whatever. Keep this in mind. Remember, we said all cells come from other cells, and another thing all cells do. They all must exchange material with their environment. And the membrane controls that. Hydrophobic molecules, non-polars, like hydrocarbons, can dissolve in the lipid bilayer and pass through the membrane rapidly. Some of the worst toxins that we have, we used to do genetic testing upstairs, and when we would do gel electrophoresis, there was a dye that you would dye the DNA with called ethyl bromide. Ethyl bromide is one of these non-polar substances. You don't have to breathe it in. Guys, you're awful interested in what's going on in the computer back there. I hope that we don't have to get to the point where we tell everybody to turn them off. Ethyl bromide is non-polar. You get it on your skin and it's in your system, spread almost instantaneous. There are other chemicals like some of the pesticides that are out there. These type of chemicals, you don't have to take internally, you don't have to breathe them in, you get them on your skin, when you're done. So you really have to be cautious. That's why you're all the time wearing gloves when you're in the lab, your face masks and all of this stuff to prevent getting any of that on you. Now notice it says water is polar, so are things like sugars. They can't get across that membrane because of the fat layer. Yeah. But you have.
Oh, I was wondering, how dangerous is ethyl bromide? What can it
Pretty
cause?
late. Well, basically it breaks down your nervous system and will eventually work on your kidney or liver.
Now, similar concept to how that works, like the skin contact thing. There was this fear that fentanyl did the same thing, but that was just recently shot down. Is it true?
I have no clue about it, what it is. I mean, I know what it is, but I don't know. You know what fentanyl is, don't you?
Yeah, it's an opioid.
No. It's a knockout drug for large animals,
Yeah, elephant tranquilizer.
what it is. So, that should tell you right there, it's not real smart to be taking it in your system.
No.
Channel proteins, aquaporins, those right there that it does eat in. Aqua referring to water. These are the proteins that are going to allow the passage of water. You can have, like we say, proteins that allow things in, carries things in and out. I don't want you to spend too much time with those. I do want you to understand diffusion is just the tendency for water to spread out evenly, just the tendency for water to spread out evenly. This shows if you put things in water that it tries to just equalize itself out. The terms I really want you to remember now. The terms I really want you to remember, notice it says substances diffuse down their concentration gradient. When something is just doing this just by its own, that's called passive. There's nothing forcing it. I want you to remember, osmosis is just the term for diffusion across the membrane of water. This shows how things will even out. Here's what I want you to remember, these terms right here. Tonicity is the ability of a solution to cause a cell to gain water. In an isotonic solution, everything is equal, both sides of the cell. Now, this is not referring just to a membrane anymore, now it's referring to the cell. If it's isotonic, no net water movement occurs, everything's equal. In a hypertonic, one part has a solute dissolved in it, and it says the solute concentration is greater than that on the inside of the cell. So the pressure outside is greater than the pressure inside. Notice what happens when that occurs. The inside of that cell membrane that has water is going to give that water up and try to go diffuse that heavy concentration on the outside. If it's a hypotonic, it means the concentrations is less than inside of the cell. So what happens? We've got a cell with a certain concentration. The concentration is less on the outside. So since water is going to try to diffuse and equalize itself, the water from the outside of the cell will move in and cause the cell to rupture and break from too much water. Remember, it says substances diffuse down in concentration and break. In other words, if it's more concentrated here, the water is going to move to where it's more concentrated. Always. Water will move to where the concentration is greatest. In a hypotonic solution, it's greater outside, so the water loses, or the cell loses water, dries up. The opposite for hypotonic, this shows with the blood cell. In a hypotonic, solution is greater, concentration is greater inside of the cell than out. So water moves into the cell and it ruptures. Isotonic, it stays normal. In a hypertonic solution, the concentration is greater on the outside. So the cell loses its water and shrivels up. And I do want you to remember this term right here. In plants, whenever that cell loses water, it's got a cell wall. Well, when that cell shrinks, that cell wall is not going to shrink. What happens is the membrane inside of that plant cell is going to pull away. Notice right here, it pulls away from it. That will kill the cell if it does too much. That is called plasmolysis. If you've ever seen a plant, whenever it's in a dry time and it starts to lose water and they start to wilt, that's because it's going through plasmolysis. The internal membranes are shrinking away. There's not enough water to make it rigid the way it's supposed to be. And that's why plants will. If it goes too far, it will kill it. That's plasmolysis. So remember that. That is the main part of this chapter that I wanted to cover. Next time, we'll talk a little bit about this active transport, finish up, and then we will start with energy.
Dr. Preston, were you saying that water and sugar, you said they're both polar? Okay, and so that's why sugar dissolves in water, right? And you were saying that water can cross the membrane, but sugar cannot by itself, right? It cannot diffuse.
Water can't go across it.
Oh, right, it has to be transported.
Got like
Sorry?
got those little